Thank you very much, Marta. And thanks to the entire PicoCon team for making this possible, for inviting me to give this webinar here. So then I'll move on. And what I would like to do today is actually different from what I usually do when I give a talk. Usually when I give a talk, I like to focus on one particular system or technique. But I thought that for this audience, maybe it would be interesting to really take this opportunity and show a little bit the toolbox that we have available nowadays for probing intrinsically disordered proteins using single molecule spectroscopy. And I mean, many of the techniques that I will showcase today are, of course, not only applicable to intrinsically disordered proteins, but I decided to choose the context of intrinsically disordered proteins because it allows us to really explain some of the strengths of single molecule spectroscopy that come out particularly well in the case of intrinsically disordered proteins. And so I'll not go into depth for most of the aspects that I will talk about, but I will, there will be sort of 4 sections in my talk, and whenever I'm done with one of these sections, that would maybe be a good time to discuss a few of the things if you have questions in more detail. Now, I will not talk about intrinsically disordered proteins very deeply from a conceptual point of view, but I would like to at least make sure that every one of you knows what intrinsically disordered proteins are, just briefly. And I assume that many of you already know what intrinsically disordered proteins are, but let me just illustrate briefly what I mean when we talk about IDPs or intrinsically disordered proteins. This is one of the most famous examples of an IDP, the tumor suppressor P53, which is important for a myriad of biological processes. And you can see here in gray is sort of a well-structured part of the protein. And this part binds to DNA, which is shown here in purple, a little piece of DNA. But then this protein also contains large disordered regions shown here in color, which traditionally were ignored in the structural biology field because if you have these disordered parts, proteins don't crystallize and they don't behave the way they're more difficult to treat biochemically. And so it's a topic that was ignored for a long time. But for the last 10, 20 years, it has become clear that these intrinsically disordered regions, where some proteins are entirely disordered, are actually functionally very important and cannot be ignored. And this tumor suppressor P53 is not a rare example, but there's a large number of proteins that are intrinsically disordered, and current estimates are that about 30 or 40% of the human proteome contain large intrinsically disordered regions of more than 30 or 40 residues or entirely disordered. So it's really a phenomenon that we find broadly in biology, and so investigating it is a very important current topic. Many of these proteins are involved in interactions with other biomolecules, be it DNA or be it other proteins. And so if we stick with the example of P53, there is more than 100 targets, binding partners known for P53, where these interactions take place via disordered regions of the protein. Right? So that just illustrates how important these proteins are for interactions in the cell. And that's one of the key biological motivations for studying these systems and for trying to get a better understanding of how they behave, how they do their function, and how they integrate in the rest of molecular biology. And so I will not go into the biology at any depth, but rather what I would like to do today in these 4 sections is tell you a little bit about the challenges of studying IDPs and how single molecule spectroscopy can help to do that. And so the first aspect, of course, is if we have such disordered systems, we can no longer speak of a single well-defined structure. Instead, we have to talk about these systems in terms of ensembles of molecules, right? So that's the first challenge. We need to describe these systems quantitatively in terms of these conformationally diverse ensembles. Then we also have to look at the dynamics of these systems, right? You can imagine if you have such a disordered set of configurations, then there's pronounced dynamics going on in these IDPs. So that's a second challenge. And so this would sort of describe the behavior of these IDPs more or less by themselves. But of course, we're also interested in their functional properties. So that would be a next challenge. And then finally, ideally, we would like to study these systems in their natural environment. So we would like to understand how do they behave in complex environments. And so I would like to touch on these four aspects. I'm not sure I'll be able to cover all of them. Maybe I'll have to skip a few things at the end. It also depends on how many questions there are. Okay, let's start with the conformational ensembles of IDPs. And so I will essentially, some of the experiments and results and techniques I will mention date back a decade or so, but then I'll move on to more and more recent results during the talk. Now, another important aspect that I would like to take along as I speak is the aspect of time. And so this is an observation or a fact of biomolecular systems that many of you probably are aware of, which is that we have to cover a very broad range of timescales, right? So we go from very rapid dynamics and the nanosecond timescale. Some people would even say you have to go to longer and to shorter timescales in the picosecond range or even shorter. Microsecond range, where you have folding and binding processes, millisecond range, that's sort of where many folding and binding processes take place, but also in the seconds range and slower, we have cellular processes, and we also have very slow processes like protein aggregation. I will not talk about protein aggregation, but in this series, for instance, you heard from Liz Rhodes, who's also been studying aggregation, for instance, with single molecule experiments. These are processes that can take place on timescales of human lifespan, right? So we really have to cover a very broad range of timescales. And over the last 25 years, essentially, we, and if I say we here, I mean the single molecule community as a whole, has developed a broad range of methods that allow us to probe these different timescales. And we will sort of look at some of these methods during this talk and how we can cover different timescales for these types of systems. I would like to start out with an approach, which is one of the classical ways of looking at single molecules, which is to measure transfer efficiency histograms, as they are shown here, and that virtually all of you will probably be familiar with. And so I'd like to take this opportunity to just mention briefly what kind of instrumentation we use without going into much detail. But for all of the experiments that I will be telling you about, we used confocal single molecule detection. And in most cases, we use such types of 4-channel instruments. Of course, the key point for looking at things like single molecule FRET is that you can separate the colors, so we can detect donor photons and acceptor photons. But for some aspects that I'll come to later on, it's important to also sort the photons by other properties, for instance, by polarization. And it can be important to have multiple detectors for donor photons and for acceptor photons for correlation spectroscopy, for instance, that I will talk about later on. And so in all of the experiments, what we have is a small confocal volume. And in the simplest experiment, we just have these molecules that we're looking at fluorescently labeled in solution, and these molecules diffuse freely. And if we look at them at sufficiently low concentration, the probability of having 2 molecules in the focus at the same time is negligible. And that's how we achieve a single molecule experiment. And so we can look at the fluorescence from these molecules as they diffuse through the focus. And this is illustrated down here. I hope you can see this move where you see down in green here the donor photons, and in red the acceptor photons. This is a little animation that was made by Hagen Hofmann. And you can see that most of the time we have very little fluorescence. That's the background that we're dealing with. And every off and on, we see these bursts of photons, which we then can analyze, for instance, in terms of transfer efficiencies, and plot these familiar transfer efficiency histograms, which illustrate already one of the key strengths of single molecule spectroscopy, which is that they allow us to resolve heterogeneity in molecular systems. So for instance, here in this case, we have unfolded protein molecules and folded protein molecules, which coexist at the same time in the solution, right? And so we can see these subpopulations and analyze them individually without ensemble averaging, right? It turns out that it can also be very useful to add additional information. And in this example, for instance, if we add information about the fluorescence lifetime, we can do these kinds of two-dimensional types of spectroscopy that Klaus Seidel and Shimon Weiss and others introduced many years ago, which we are using extensively to study proteins and intrinsically disordered proteins. And so from this, we can get information about the subpopulations, as I mentioned. And so, for instance, here, these are experiments done under different experimental conditions where we add denaturant like guanidinium chloride, and at low denaturant concentrations, we just have the folded population. At high denaturant concentrations, we increasingly populate the unfolded state. And you can see the two populations, but you can also see that the unfolded state changes as you change the solution conditions. And this is one of the first results on protein folding that we obtained using single molecule experiments. But more generally speaking, what we can get from these types of experiments is information about the distances and distance distributions within the molecules, right? If we use single molecule FRET, then we can extract distance information between our fluorophores. And from the average transfer efficiency of such a peak, for instance, we can infer the average distance between the two fluorophores. And in addition, if we also use fluorescence lifetime information, we also get information about the width of these distributions, because it turns out that if you have a distribution of distances that rapidly sampled, our population does not sit on this diagonal line that you expect for a static system, but it deviates from this line. And this deviation here is proportional to the variance of the distance distribution. So in this way, we can get information about mean and variance of this distribution. We can describe these distance distributions in terms of, for instance, simple polymer models. Of a favorite polymer model is the SAW-NU model that we developed with Wenwei Zhang and Robert Best at the NIH. But if you have simulations, for instance, then you can also use those to compare your single molecule experiments and the distance information and the distributions that you extract. So that's one piece of information that we can get. And let me just quickly skip this and mention briefly a few examples of the type of experiments, the type of information about IDPs that you can get from these types of measurements. And so these are some of our early experiments when we started working on IDPs more than a decade ago. And so, for instance, if we take a highly charged, intrinsically disordered protein like this one here, prothymosine alpha, which I will come back to later on, which is a highly negatively charged IDP, you can get information about the average dimensions of this protein from the transfer efficiency. And we can see now here, if we plot here something like the apparent radius of gyration of this molecule, if we add salt, like here a molar of potassium chloride, or a charged denaturant, then you can see how the Rg drops. So that means you start with a relatively expanded chain, and then you add salt that screens the charges, and the chain becomes more compact. And then we can use concepts from polymer physics, for instance, polyamphilide theory, to describe this quantitatively. And so this is work that Andrea Sorano, together with Sonja Müller-Speth, pioneered in the lab. And Hagen Hoffmann did a lot of work in the lab on these kinds of questions. We can really probe very systematically the dimensions of intrinsically disordered proteins, but not only as a function of salt, for instance, but also as a function of temperature. And so one of the surprising results here is that in contrast to what you might expect, if you take an unfolded protein and you increase temperature, then the dimensions of the protein actually decrease. So proteins tend to become unfolded, and disordered proteins tend to become more compact as you increase temperature. And together with Robert Best, who did extensive simulations on these systems, we found out that what you need to take into account is the solvation-free energy of these molecules. And actually the temperature dependence of this solvation-free energy is what explains this compaction of intrinsically disordered proteins. And the last example I want to mention of these types of measurements of dimensions of IDPs is a more complex system, just to show that we can also use get information about more complex systems. And so here, what we looked at is the interaction of a molecular chaperone, DNA K and its co-chaperones, but in particular DNA K, with a disordered, or in this case, a non-native protein, a protein that's not yet folded. And so what these chaperones do is they bind to the unfolded protein shown here in color. And so here one chaperone molecule is bound, but multiple chaperone molecules can bind to this chain. And you can see as multiple chaperone molecules bind, and these are simulations done by Alessandro Barducci, the unfolded protein gets more and more expanded, of course, because these chaperones are large and they repel each other because of their excluded volume. And so this can also lead to a change in dimensions of unfolded proteins. And this is also something that we can look at with single molecule FRET. And so this was sort of a brief introduction to the ways we can probe these ensembles, how we can probe distance distributions in IDPs. And maybe before we move on to the next section, which will be about the dynamics of IDPs, I could take any questions if there are any. No questions so far? Please don't be shy. I think it's great that we have this means of discussing a little bit, even if it's virtually.
So Ben, in fact, we have one question.
Yes.
Yeah. So in this single molecule experiments, in terms of the proteins for the fluorescent labeling, do you what would be a best label, like labeling with GFP, like fluorescent protein, or small molecules?
Yeah, so that's something I should have mentioned. You're right. We use small fluorophores, or as small as they get in the visible range, because GFP is, on the one hand, it's relatively large, actually pretty large compared to these intrinsically disordered proteins that we typically probe. GFP is a pretty large protein itself. And so it would be a little bit like chaining a cannonball to a piece of wool. And secondly, the photophysical properties of GFP are not quite as good as those of these small organic fluorophores. And so we use these kinds of fluorophores, and this example, for instance, Alexa 488 and 594. Another pair that we like to use is Cy3B and CF660R. And we typically attach them via cysteine residues that we introduce into the chain. And it turns out that for many IDPs, that's relatively easy to do because they rarely contain cysteines by nature. And so it's relatively easy to just put in cysteines and label them quite specifically, and then purify, for instance, by high resolution reversed phase HPLC, which gives you very good resolution in many cases. But then there are, of course, systems where you have to go beyond this relatively simple labeling, and you can use non-natural amino acids, for instance. You can use intein chemistry. There's all kinds of more sophisticated biochemistry that we use whenever it's necessary. But in most cases, labeling via cysteines is a quite reliable strategy.
Thank you.
We have another question from Luis, so you can unmute yourself.
Yes. One question. So it's about the labeling. So when you said that you add cysteines, but for this you need to add two different groups. So then how you define which cysteine to be modified at which time?
Okay, so how to choose the positions, essentially, that's what you're asking, right?
How to choose the position and how to protect the other cysteine that because you said that you need to modify to add two cysteines in a way that they will be modified. So then how you modify one with one group and then the other with a different group?
Okay. Okay, so these are two questions. One is where to place the dyes. And so typically what we would do, so for instance, this example, this is a protein that actually folds. And so you would place them in a way that they don't interfere with the folded structure, right? Or if you have you investigate an enzyme, say, then you don't want to put the dye into the active site because that will interfere with its activity or binding of substrate and the like, right? So on the one hand, you need to place them in the way that the process you want to investigate and the type of structure and dynamics and function are not perturbed more than absolutely necessary. The second question is how to get them labeled. What we typically do is you have the two cysteines. If there are natural cysteines, you either have to take them out or you have to find a way of protecting them, right? But in most IDPs, the number of natural cysteines is quite low. So in many cases, we can just put in two cysteines and there are no natural cysteines. And then we typically add one fluorophore. We purify that using chromatography and purify out the singly labeled protein, then add the second dye, and then we have donor acceptor labeled protein. And in many cases, it actually turns out that the chromatography is good enough that you can also separate the permutations. And in this way, we get quite specific labeling. Okay. Any other questions? If not, then let me move on to the...
Sorry, sir. Sir, I have a question.
Oh, yeah.
IDPs had not a particular structure like in ordinary protein structure, have fixed 3D structures in ordinary proteins. So so it is any problem in labeling of IDPs in comparison to normal proteins which have fixed 3D structures?
The basic procedure is the same. Actually, in many cases, it's a little easier because for folded proteins, in many cases, you have to sort of make sure that you can keep the native structure intact. And in IDPs, you can purify them under highly non-native conditions. You can use organic solvents. You can use all kinds of conditions, which simplifies things because it increases the choice of chromatography that you can use. But fundamentally, it's very similar. Whether you label IDPs or folded proteins, the procedures are closely related. Yeah. Okay, so then let's move on to the second part where I'll talk a little more about the dynamics. And so in this case, what we're particularly interested in for intrinsically disordered proteins is the dynamics of the entire chain. How quickly do different configurations of the chain interconvert? This is what we call the reconfiguration time of these molecules. And it turns out that we can measure this quite efficiently using nanosecond FCS, which is a variant of FCS, fluorescence correlation spectroscopy, where you sort of extend the timescale down to the nanosecond and even picosecond range. But it turns out that the dominant timescale for many IDPs is sort of in the timescale of tens to hundreds of nanoseconds. And these are such correlation functions. But let me show you in a little more detail how this works, right? So the basic idea now is we do not just want to get information about the average distance between molecules, but we want to get information about the fluctuations in distance between our two fluorophores that we have attached to the molecule, right? And so these fluctuations we can measure using correlation spectroscopy. And this is an example of one of the first correlation functions that we obtained at the time. And Daniel Nettles pioneered these experiments in the lab where we measured this unfolded protein, and this decay component here on the timescale of about 40 nanoseconds reports on the distance dynamics between the two fluorophores. There's another contribution here at very short times. This is just photon antibunching, sort of the fundamental quantum mechanical behavior of a single molecule that we expect, but this is the timescale that we're interested in. And if you have such a flexible disordered protein, you see this component. If you use a stiff molecule like this polyproline peptide here, for instance, this component is absent and the curve is completely flat, essentially, right? So this shows you that you're really probing distance dynamics. And so this enabled us to really more systematically explore the dynamics of intrinsically disordered proteins and unfolded proteins and compare them to concepts from polymer physics, which also make predictions about the dynamics of polymers. And so to do this, we now essentially have to bring together the information from the first part of the talk, where we got information about the distance distribution, with the information from nanosecond FCS, where we probe the dynamics. And what's shown here now are the three correlation functions that we can get from a single measurement, right? We can autocorrelate the signal, the fluorescent signal from the donor. That will be the correlation function in green here. We can autocorrelate the signal from the acceptor. That's the red correlation function. And we can cross-correlate the signal between the two. That will be the blue correlation function. And the key signature that you're looking for if you have distance dynamics, of course, is that the cross-correlation function should have a negative amplitude. Or in other words, the emission of the donor and the emission of the acceptor should be anti-correlated, right? Because if the two fluorophores are close, you expect a lot of emission from the acceptor. If the two fluorophores are far apart, you expect a lot of emission from the donor and low emission from the acceptor, right? So they are anti-correlated. And this is what you can see here directly in the cross-correlation function. And so this is a key signature for distance dynamics. And all of these three correlation functions need to show the same decay time. And then we can be quite sure that this is distance dynamics that we're talking about. And now the nice thing is we can combine these two pieces of information, the distance information from the experiments that I told you about before, and the dynamics information from the nanosecond FCS. And for this, we use a concept or a theoretical framework developed by Irina Gopic and Attila Szabo to calculate these correlation functions. And the basic idea, without going into much detail, is that we use the distance distribution here. Then we convert this distance distribution into a potential of mean force, right? Just by taking the logarithm of this distance distribution, we get a potential of mean force. And now we treat the dynamics of this relatively complex system in terms of simple diffusive dynamics in this potential of mean force. And we can, in this way, get information not only about average quantities that describe the distance distribution, like the mean squared end-to-end distance, or the radius of gyration, or the scaling exponent, if you like, in terms of polymer physics, but also in terms of a reconfiguration time. That would be sort of the characteristic time scale with which different configurations in the unfolded state or the disordered state interconvert, or an effective diffusion coefficient in this potential, right? So this would be a very simple description, if you like, in terms of diffusion. We only have a single degree of freedom here, the end-to-end distance, and we can describe it with the simple diffusion equation in this potential. But one of the wonderful things of the developments of the last decade or so is that molecular dynamics simulations of these kinds of systems have improved to a level that we can also do a direct comparison to a much more complex model, right? So here we describe really the whole system in all of its beauty. What's shown here is really just the polypeptide chain, but in these simulations, we have an atomistic representation of all of the amino acids in this protein. We have water molecules included explicitly, and the simulations can now get long enough that we can really do a direct comparison of experiment and simulations. This particular example is from simulations of Stefano Piana and D.E. Shaw's group. And it turns out that for most of these systems where we now have a quantitative comparison, the timescales that we get from our experiments agree remarkably well with these molecular dynamics simulations. And I'll come back to a more specific example in a few minutes. And so maybe I'll skip the next slide because I see that we're already much slower than I anticipated. And so let me show you a very recent experiment and some simulations. And this is work by Mark Nirsch in the group and Mila Ivanovic, and a collaboration with Jerome Wenger in Marseille, and Robert Best at the NIH. A lot of help from Daniel Nettles. And this addresses two of the current limitations of this nanosecond FCS, as we call it. One is that dynamics that are faster than about 10 nanoseconds are very difficult to resolve, because then we start to overlap with this photon anti-bunching, right? So maybe if I just briefly go back to one of the correlation functions, so you can see it here, right? If the dynamics are very fast, then they start to overlap with this anti-bunching peak here, and then they become very difficult to probe. That's one limitation. The second limitation is an aspect that I haven't mentioned, which is that these measurements typically take a very long time. So one of these correlation functions typically takes data acquisition of about 10 hours to really have sufficient signal to noise. And so what we did in a collaboration with Jerome Wenger is to use his zero mode waveguides to change the situation. And zero mode waveguides, some of you may know, they were introduced about 20 years ago by Webb and collaborators, especially to be able to do single molecule experiments at higher protein concentrations. Because as many of you probably know, if we just do a standard confocal single molecule experiment, typically have to do these experiments at picomolar concentrations, or maybe nanomolar concentrations if you do FCS. But if you want to go to higher concentrations, you lose the single molecule signature. And so to reduce the observation volume, you can use zero mode waveguides. And what are zero mode waveguides? Essentially, they're just little holes. And so what Jerome Wenger and his co-workers do is they deposit a thin film of aluminum on a glass slide, and then they microfabricate or nanofabricate little holes into this layer of aluminum. This is an electron microscope picture of such a zero mode waveguide. And then what you have is an evanescent field here if you illuminate this from the bottom. So essentially we just place our confocal volume here, and that means that we just illuminate this region. And what happens is that you have an enhancement of the electromagnetic field in these zero mode waveguides. And this is one important effect. By this enhancement, we get more photons, and that increases the signal to noise by about an order of magnitude. And the second effect is that the lifetime of the fluorophores is decreased in these zero mode waveguides. And so that means that the anti-bunching peak becomes narrower, and we can probe faster dynamics. And so this is shown here with an example of a short disordered peptide that Milos and Mark have been looking at. And so these are measurements in the zero mode waveguides on the right, and without zero mode waveguides under comparable conditions on the left. And you can see that here it is very difficult to get any information out. It's just much too noisy. But in the zero mode waveguides, you get beautiful correlation functions. And down here, you can see a comparison of a 20-hour measurement with a classical confocal measurement. And here on the right, measurements of about 10 minutes in the zero mode waveguide. And you can see you achieve comparable signal to noise, right? So that really allows us to do these measurements much more quickly. And especially if you now look here at the anti-bunching behavior of the acceptor signal, you can see that it becomes much narrower here in the zero mode waveguides. And so we can resolve dynamics in the sub 10 nanosecond regime. And Mark has gone down to dynamics as fast as 4 nanoseconds. And another great aspect of this is now we can compare this directly to molecular dynamics simulations done here by Milos in the group in collaboration with Robert Best, who developed over the years or optimized and adapted force fields that enable faithful simulations of intrinsically disordered proteins and explicit fluorophores, which we can now include here directly. And so we can directly compare experiment and simulation by also simulating now photon emission from this type of system. And we can get information about the distance distributions in these systems and directly compare, for instance, the correlation functions here on the left, the correlation functions measured experimentally that I just showed you. And these are now correlation functions from photons simulated based on the MD simulation. And you can see that the agreement is actually quite remarkable. And so this tells us that we now have tools both from the experimental side and from the simulation side to really get a much more detailed understanding of the dynamics of intrinsically disordered proteins. And so with this, that sort of completes my section on the dynamics of IDPs. So if there are any questions here, maybe we can discuss those before I move on to the function of IDPs.
Hi, Ben. So my question is about the nanosecond FCS.
Yeah.
What exactly, why does the data acquisition need to be as long as 10 hours to get one curve there?
Yeah, so the key point is that if you think about the average interphoton time, or the average number of photons per time that we get in such an experiment, that's on the order of, say, a photon every 10 microseconds, right? So if we now want to probe dynamics on the nanosecond timescale, then we need to have photons that are spaced by times in the nanosecond range. And these are just relatively rare events, right? So if the average interphoton time is about 10 microseconds, right, then the probability of having photons that are spaced by a few nanoseconds is just very low. And that's why we need to accumulate a lot of data to be able to have enough photons that are separated by nanoseconds or less. That's the main reason.
Okay.
It's just
thanks.
statistics.
Yep, thanks.
Hi, Ben. If you have shorter lifetime probes, in that case, can you go down to, let's say, 2-3 nanoseconds? If we have very 1 nanosecond or less than that lifetime probes.
Yes, in principle, if you have fluorophores that have shorter lifetimes, you can also go down and probe shorter dynamics, faster dynamics. Absolutely. It's just the question of whether these fluorophores then still have a sufficiently high quantum yield, for instance, that they emit enough photons in the first place, right? But ideally, you would have a fluorophore that has a very short lifetime and a very high quantum yield. Unfortunately, this combination is relatively rare. But in principle, that's exactly what we would like.
Hi, yes, just want to know if the same approach could be used to study protein-protein interaction. So not like intramolecular interaction, but with, for example, two proteins that will interact and change conformation in a time frame like it's very fast. So would this method could be used for this?
Yes, maybe we can discuss this question in the next section, because I'll come exactly to examples of this type. Yeah, when we talk about the functional properties, where interactions between IDPs are, of course, one of the key functions that we would like to probe and need to probe to understand the behavior.
Yeah, hi Ben, nice to see you here.
Christian.
So could you briefly comment on how you passivate the zero mode waveguides to avoid sticking of your stuff to the waveguides?
Yes, you're right. I mean, you're of course a specialist also on surface interaction, so you know exactly this is a key point. And so what we do, or what Jérôme does, or what Jérôme told us how to do it, is to just put a very dense peg brush on the surface. This does a pretty good job for most molecules. There are still some highly positively charged proteins, for instance, that are so sticky that they're still problematic. And so we're still in the process of further optimizing surface passivation, but a dense peg layer does a pretty good job for most systems.
Okay, thank you.
Okay, so then let's move on to function. And so here I would just like to briefly show a system where you've looked at interactions, which are also quite remarkable from the biomolecular point of view. And so here what we have is, again, this highly charged protein that I mentioned before by the name of prothymosine alpha, which we labeled here with a donor and an acceptor. And this would be the transfer efficiency histogram, where you get a relatively low transfer efficiency considering the sequence separation of the dyes. Because this protein is so charged, it's relatively expanded. But now you can add a binding partner, and it turns out an important binding partner is the histone protein H1. And so this histone, if you add it, even at picomolar concentrations, you see here a second population that corresponds to the bound complex between the negatively charged prothymosine alpha and the highly positively charged histone H1. And so that already tells you that the affinity between these molecules is in the picomolar range. So these two proteins bind to each other very, very tightly. But we would also like to get more information about the properties of this complex. And this is where FRET turned out to be an ideal way of doing this. And in a tour de force from the biochemistry side, Alessandro and Madeline Borgia managed to place the fluorophores into several different positions, both in this histone H1 shown here in blue, indicating sort of its positive charges, and prothymazine alpha, the negatively charged guy shown here in red. And so every combination of a position here and a position here gives you one transfer efficiency histogram and one transfer efficiency. And that sort of gives you information about average distances between different points between these chains. We can, of course, also do this within the chains. But if you just look at these transfer efficiencies, you cannot sort of come up with a simple arrangement that would fit all of these transfer efficiencies. And so again, this is an example where simulations turned out to be very useful. And so Robert Best did these simulations. These are now not atomistic molecular dynamics simulations, but coarse-grained simulations. But still, we can compare these to the experiment. And so you can calculate transfer efficiencies from these simulations for all of these different distance pairs that Alessandra and Madeline prepared, and compare the experiment, that's the squares, to the simulation. And it turns out if you tune the simulation accordingly, then you can describe this experimental system pretty well. And so this is what we think this complex actually looks like. And you can see it looks very dynamic. These proteins actually do not form any structure when they bind to each other, as we know from CD and NMR. And you can also probe the dynamics in this complex, right? And so we can do again our nanosecond FCS experiments now for the complex between H1 and prothymazine. And you can see here now from the correlation functions that the dynamics are still very pronounced, and they're very similar to the dynamics of the proteins in the free state. So we really have a very dynamic, disordered complex between these proteins. And that's interesting from a biological point of view, because it's sort of an extreme case of disorder in the protein complex. So here on the very left, you would sort of have the textbook example of a protein-protein complex. But we now know from research over the last decade or so that there is different degrees of disorder in protein-protein complexes. And this would be sort of an extreme case of disorder in such a complex. And this is not the only example. There have been more of these cases now. And we summarized this here in the review last year. Okay, but all of the experiments that I talked about so far did probe the behavior of these molecules at equilibrium. And I see that I'm already running out of time. So let me just mention this briefly, that in many cases, it is actually useful to do non-equilibrium experiments. And here we can use microfluidic mixing. And this is a technique that we've been using for quite some time now. It was the very beginning was in a collaboration with Everett Lipman when I was still a postdoc in the lab with him together in Bill Eaton's lab. And then we developed this over the years. And we can now sort of rapidly mix molecules in these microfluidic devices and then probe their behavior in such non-equilibrium kinetics. But let me just jump the next slides, because I would like to show you a recent development that Tianjin Yang and the lab has done, where she now developed a droplet-based microfluidic mixing system. The idea here is that you mix your samples. So you have two samples that you want to mix, say two proteins that you want to bind to each other, and look at the kinetics of binding. And then they get mixed here and immersed into oil. And so they form a little droplet. So you have a water droplet in oil, which then moves down this channel. And because of the serpentine channel, there's sort of chaotic advection going on in the droplet that mixes the solutions very quickly within a millisecond or so, or a few milliseconds. And then you can look at these droplets in the channel. And here's actually a movie of such a device that Tianjin fabricated and tested here. And you can see, I hope you can see the droplet move down the channel, and that it's slowed down here so that we can look at it using confocal detection. And then, for instance, measure the kinetics, the kinetics of protein binding. Here again, for the example of this positively charged H1 binding to prothymazine alpha. And this is an experiment that was previously not possible because H1, this comes back to Christian's question, is an extremely sticky molecule. And so if you use this in regular microfluidics, it'll just stick to the surfaces and you'll never see it again. But here in these droplets, we can prevent surface interactions. And so we can measure even such challenging systems using microfluidic mixing. And so this takes me to a different type of experiments. So all of the experiments that I talked about before were on molecules that were free in solution, either at equilibrium or in microfluidic mixing devices. But of course, there's a whole set of different types of experiments where you use surface immobilization to probe these experiments. And that's also something that's very helpful to probe the function and the interactions of IDPs. And the basic idea is very simple, as shown here. So you mobilize one of these molecules, say, on the surface, and that goes back to the question that we discussed about intermolecular FRET and interactions. So here you would have one molecule sitting on the surface with one of the dyes, the donor in this case, and other molecules freely diffusing in solution with the acceptor. And then if this molecule is by itself, you expect donor emission. And if the binding partner binds, you expect to see an increase in acceptor emission. So you would expect to see a trajectory like this. We start out in the free state, we bind, the acceptor emission goes up. Then the binding partner dissociates, acceptor emission goes down. And so you see this typical anti-correlated behavior of donor and acceptor emission. And so this is just a short snippet, but you can actually measure very long trajectories of up to several minutes. And you can see now here, this is really hundreds of binding events recorded from a single donor labeled molecule. Of course, the acceptor labeled binding partners are always different ones, but you can get from such a trajectory, you can probe the kinetics of these molecules, even from a single trace at quite remarkable detail. And so just one example that I want to mention is that from these trajectories, you can actually see kinetic heterogeneity that would be difficult to pick up in ensemble experiments. So this is the same kind of experiment. Now, the other protein partner is actually immobilized on the surface. And if you now look at this long trajectory here, you can see that there are two different regimes. One that's sort of underlaid here in pink, where you can see that there are relatively long-lasting binding events here. Some of them last for a few seconds even, right? But then you have this other regime that's indicated here in green, where you only have very short binding events. They're blown up here, and you can see that they're much shorter. And so there's really two different types of behavior, even though we're looking at a single immobilized molecule here that's sitting on the surface. But it turns out that what's going on here is that in this molecule, you have a process that's called proline cis-trans isomerization. So one of the peptide bonds, and that of proline 20 in particular here, is actually changing between a cis and a trans configuration. And the behavior of this molecule is different in these two configurations. And so it binds more tightly in one than in the other. And we can see this here directly in the trajectories, where here in this pink stretch, it's in one isomer, and in the green stretch, it's in another isomer. And if you do a quantitative analysis of these data in terms of dwell time distributions and do more detailed hidden Markov modeling that take into account the kinetics, we can extract the kinetics of the system in remarkable detail. So that just as an example to show you that we can get information on relatively long timescales and on relatively complex kinetics from single molecule experiments. Now, I don't have the time to go into this in much detail. Let me just mention that you can also look at very rapid dynamics, even from these kinds of trajectories. And these are experiments of what I told you about before, are experiments by Franziska Zosel and Florin Sturzenegger in the lab. He looked at these actual transitions in more detail, and turns out from those kinds of experiments, you can get information about transition paths, so the actual time it takes for molecules to go from an encounter complex where they start to see each other to a fully bound state, and that occurs on the time scale of tens of microseconds. So you can really also with these experiments probe very rapid dynamics. But I think I'll have to skip that because I'm seriously behind in time, and maybe we'll have to finish here. But maybe if there are questions on how we probe functions of IDPs, we can address them here. Are there any questions? No? Maybe I was already moving a little too fast here. but we are essentially at the end of the time that I have. So let me just mention very briefly, without too many details, that one of the last challenges that we have is to probe these molecules in a complex environment, because of course in nature, it's not just these molecules by themselves, but there are other molecules around. Ideally, we would like to look at them inside the cell. And so this is one of our biggest challenges, is really to understand these molecules in a more complex environment. And let me just highlight this example here, which will soon be published. This is the interaction of this histone H1 that I mentioned now with a large complex, the nucleosome, which is the key structure in the cell's nucleus, where the DNA is wrapped around, the blue here would be the DNA wrapped around the core histones. And this histone H1 here can bind to the nucleosome, and we can probe these interactions using similar kinds of experiments as I just showed you. We can immobilize the nucleosome here and then measure binding of H1. But then we can also look at the effect of prothymazine alpha, of which I showed you that it interacts with H1, and see how it affects the nucleosome. And it turns out that if you analyze these kinds of experiments quantitatively, what is going on here is that prothymazine alpha actually can bind to the histone H1 while it still sits on the nucleosome. And so essentially what it does is sort of sneaks into the nucleosome, right? So here you have histone H1 bound on the nucleosome. Prothymazine alpha comes in and binds to this complex. This is relatively unusual that you form these kinds of ternary complexes, and then it speeds up the dissociation of H1. And so this is an example of the interaction of these IDPs in a more complex environment. Ideally, we would like to do this in the cell, and we can mimic the cellular behavior using crowding agents, but we can also do such experiments now in live cells. So what we do is we just micro-inject these molecules into the cell, and these are experiments pioneered by Ivo Koenig, Arash Serinov-Sar, and Mikhail Atsnourian in the lab. And then we can essentially do the same kinds of experiments. We just have to deal with background and bleaching and other complications, but we can measure bursts from single molecules. We can get transfer efficiency histograms. We can do FCS. We can even do this kind of nanosecond FCS in the cell. And so this would ultimately be a goal, of course, to be able to bridge the gap between the experiments done in vitro and experiments done in the cell to really relate the behavior and understand what's going on in the natural environment of IDPs. And so with that, let me sort of conclude in terms of the timeline that I wanted to show you. And even though it ended up being a little hectic, I hope I was able to sort of give you an idea of the range of methods that we now have available to probe protein dynamics in general, intrinsically disordered proteins in particular, from the nanosecond range to seconds and minutes. We can go to longer timescales. Also, I didn't mention this, we can go to hours, in principle to days and weeks. We can do experiments in live cells. And so this now enables us to really probe the behavior of IDPs quite thoroughly. But I would not like to finish without saying that in many cases, of course, single molecule experiments are not the whole story, but we ideally combine single molecule experiments with other techniques, for instance, NMR, or small angle X-ray scattering, or ensemble kinetics, and take all of this into account for the models that we build to try and understand how these systems behave. And then we can use simulations based on these models to recalculate experimental observations, compare with the experiment, and then we sort of iteratively go through this cycle to try and improve until we have good agreement and think we have a sufficient understanding of the system we're looking at. And with this, I would like to end and thank a lot of people. I cannot go through the whole list. I mentioned, hopefully, most of the people directly involved in the experiments that I mentioned along the way. So these are people over the years in my group. Many of these have moved on and now run their independent positions. We also have had a lot of collaborations with experimentalists like from NMR, from SACS, dynamic light scattering, EPR, nanophotonics, I mentioned, also microfluidics, and also theory and simulations. And this is really something that has been a lot of fun over the years, to combine experiment, theory, and simulation very closely. The key person here, of course, is Robert Best, who collaborated with intensely. Irina Gopic and Attila Szabo were key people for getting the photon statistics under control. Dima Makarov for many of the theoretical concepts, but many others. I cannot mention them all in detail, but I thank them all because it's been fantastic to work with all of them. And I think altogether we've taken quite a few steps towards understanding protein dynamics. And with this, I would like to thank them, our funding sources, and you for your attention. Thank you very much. And if there are more questions, I'll be happy to discuss them now in more detail.
Yes, so very nice talk. Thank you. Have you experienced working, so incorporated in this system, for example, liposomes, like proteins in a membranous environment?
This is something that we have not investigated in detail. We have done a few experiments with collaborators on membrane proteins and their interactions and their diffusion in the membrane, but this is not a topic where we have done in-depth studies. So there are other people who have looked at membrane proteins in much more detail, like Scott Blanchard, for instance, is a name that comes to mind. They've really done some fantastic work on GPCRs and quite a few other people. We haven't really worked extensively on membrane proteins or membrane interactions. Any other questions?
Yeah, Ben.
Yes.
In case of nanosecond FCS inside the cells, how do you improve your signal to noise ratio? Because there you cannot measure for a few hours or let's say 1 hour. So
Yes.
how do you optimize the system in that case?
Yeah, that is indeed quite a challenge. And so the correlation functions that I showed you, so these are some more recent correlation functions, for instance, that Ivo took. These are not correlation functions that can be taken from one single cell, right? What we need to do here actually is we need to do these measurements in multiple cells, because you can, as you can imagine, the cellular volume is quite limited, right? And so you can only introduce a certain number of molecules not to exceed nanomolar concentrations. So you can still do nanosecond FCS, but still, once all of these molecules that you injected are photobleached, that's the end of the measurement in that cell. So what Ivo had to do is to accumulate measurements from multiple cells, typically on the order of 10 to 100 cells, to accumulate enough statistics for these nanosecond FCS experiments. And as you can see, they're still relatively noisy compared to what we can do in vitro, right? So we can see the dynamics, they're clearly there, but this is not nearly the kind of quality that we can relatively easily now get in vitro experiments. So this is much more challenging, of course, to get enough statistics here.
Okay, thanks.
Yes, I'd like to also thanks for the talk. I really enjoyed it.
Thank you.
It's great. But my question is more like general, especially in the beginning when you were showing different cases for this, like dynamics and conformational changes. Were you working with some real proteins, or was it just some model peptides, and how long or big the proteins were?
Okay, so essentially all the cases I showed you, apart from the measurements in the zero mode waveguides, were done on natural intrinsically disordered proteins. Thanks for bringing this up. There's something I forgot to mention. So all of these are either disordered parts of proteins. So these disordered proteins come in two flavors, right? One type that contains both folded parts and disordered parts, and the other ones that are completely disordered, right? And so, for instance, for the proteins that I showed you that bind to each other, that actually there was this pair here. This is go by the name of actor and NCBD. These are two fragments, two disordered fragments from larger proteins that interact with each other. But another example that I showed you, for instance, the interaction of histone H1 with prothymosine, these are actually the full-length natural proteins. So these are the proteins as they occur in nature. And of course, this is a key point, right? We want to understand the natural behavior, but sometimes it can be helpful to make synthetic sequences, right? So for instance, if you want to systematically study the effect of sequence on affinity or the like, then it can be useful to make artificial sequences. But whenever possible, we work with the natural sequences because these are the systems that we're interested in. And then we can also relate it to the biology of these molecules, which are, of course, in many cases, very, very interesting.
Okay, cool. Yeah, interesting. Thank you.
Thank you.
Are there any more questions? You can just unmute your microphone and ask them directly.
No, doesn't seem to be the case.
Yeah, it
Then
seems this is the end. So thank you once again for your participation. I hope you enjoyed the time with us and have a great day. And if you have any further questions, please contact us via info@picoquant.com. And goodbye. Stay safe.
thank you very much.